This is part 2 of chapter 18, and we're going to talk about handedness right now and amino acid handedness. So this is kind of another organic chemistry review section, and we're going to talk about the difference between chiral objects. So objects that have handedness, or in other words, cannot be superimposed in the mirror image, are said to be chiral. So where, on the other hand, achiral objects or non-chiral, their mirror images are superimposable. So two examples here are your hand. This is a chiral object because you can't superimpose this here. So if I were to take this hand over here and try to place it on top of this hand, in the mirror image, right, the hand, they won't match up, right? You can give this a high five, but the palm is going to be on the back, or vice versa, or the thumb is going to be on the wrong side of the hand. Whereas this image, I can place this essentially on top of, and the images are the same. And I'll talk about that in a second, why that's important biochemically. Here are we're talking about amino acids, and this is an example of alanine, both the left-handed version, or L-alanine, or the right-handed version, the D-alanine. So the L stands for laevus, which is left, and a D stands for dexter, right. We also talked about this in organic chemistry. We have the R for rectus as well as the S for sinister or left. But here, most of the time when we're talking about amino acids, we have the L or D naming system. All of the natural amino acids that occur in biological compounds are L amino acids. Here's just an example of an organic compound. Propane over here. You can see propane, the mirror image is exactly the same. It's superimposable on itself. Whereas here, if we put this on top of, if we put the L-alanine on top of D-alanine, you'll notice that nitrogen group is on the wrong side of the molecule. So the difference between, here's an achiral molecule versus a chiral amino acid. So chiral centers, remember, these are places on an atom where there are 4 different groups that are attached. So let's look at the chiral carbon in alanine here. Notice this alpha carbon has the amine group attached to it, carboxylic acid group, a hydrogen, and a methyl group. So it's got 4 different unique chiral, 4 unique groups attached to it, and that's how we know that something is chiral, whenever it has 4 groups attached that are different and unique. Propane, on the other hand, is achiral. This carbon here has 2 hydrogens, so it has multiple identical groups attached to it. Therefore, it cannot be chiral. So we don't necessarily name these compounds based on how there's not a direction correlated with how we name them in organic chemistry in terms of the polarization of light. Pairs of enantiomers with different have a different capacity to polarize light. And an enantiomer is basically that mirror image form of the chiral molecule. So one form will polarize light or bend or turn light in one direction. The other will do that in the exact opposite direction, the equal amount. So if you have a racemic mixture or an even mixture of these compounds, when you put light through them, they actually won't be polarized. But if you can purify these 2 different forms out, you'll be able to polarize light in different ways. Why are these important in chemistry or biochemistry? They're especially important in biochemistry because, remember, all of these compounds, these amino acids, all of the things that kind of run biochemistry are enzymes. And these enzymes have different regions and pockets that recognize these compounds and molecules. For example, here we have a chiral molecule, and the receptor sites in the protein. You can see these 3 different spots here that might be recognizing that molecule, that chiral center molecule. And we see that, the square, or the spherical peg here, square peg here, and a triangular peg here. If we have the enantiomer, the opposite form, right, the mirror image form, it's not going to fit in that enzyme pocket the same. So this is really important in biological compounds. If we have the achiral, or if we have the enantiomer of that chiral molecule, they're not going to be recognized by the enzymes that are going to process them. So having L-amino acids to make up larger peptides and proteins are essential for normal function. So our body cannot use the amino acids to make proteins because of this phenomenon. And just an example of how differences between these enantiomers. So sometimes the opposite enantiomer can have different tastes. They can have different activities in terms of their ability to act as drugs. Here's a difference here, spearmint or caraway. So this is kind of like rye bread kind of flavoring versus spearmint. It's just this chiral carbon here, right? This is the enantiomer, the opposite, the mirror image form. Those two things have very vastly different tastes to us. So just as well as the 19 of the 20 amino acids, obviously glycine has 2 hydrogens attached at the alpha carbon, so that's the one non-chiral amino acid, but the other ones are all L-amino acids, and it's essential that we can only use L-amino acids to synthesize proteins. So you've all seen this. This should also be reviewed. 4 levels of protein structure. We have the primary structure, secondary structure, tertiary structure, and quaternary structure. And of course, the primary structure is just the amino acid, the covalent bonding that gives those, the variety of 20 amino acids. It's that amide bond that is basically creating that long chain and that primary structure. So it's just the initial structure of the amino acids. Here's just an example of, kind of instead of cartoon form, the actual, what the compound is going to look like. And the residues are just given residue 1, 2, 3, and 4. But we have the N-terminal side, of course, it's important, right? So the N-terminal region is going to kind of be N-terminal here, and this is the C-terminal side, and of course that's referring to the amine group versus the carboxylic acid group, and this is the newly synthesized C-terminal side. So I always like to do N to C, 5' to 3'. That means that this is reading this mRNA from the 5' end to the 3' end to synthesize that peptide. So we can see these important functional groups here. This peptide bond is actually an amide functional group, right? And these are really important functional groups. That is the covalent bond that's being formed when the nitrogen, the amine group, and the carboxylic acid group of 2 different peptides come together, right? Water is released, and then we have this new amide bond formed. Primary structure is just that sequence of amino acids. The secondary structure is the backbone hydrogen bonding. So this is all, there has no influence, the side chains have no influence on alpha helices or beta sheets. It's all the backbone hydrogen bonding. So that's the good way to remember that, and kind of differentiate it from the different types of structures. These result in alpha helices and beta sheets. You kind of see the difference here. You can see these on the bottom, these pleated beta sheets, and they're long. They're kind of shown this way because of the structures they form. They form generally these long sheets of of peptides, where the alpha helices actually kind of do a turn, they do a spin, and they create these more of these tube kind of helix shapes that you'll notice and remind you more of like the DNA. Again, only the backbone of the chain is making the hydrogen bond. So the nitrogen, one of this amide, right, bond, this nitrogen has a hydrogen attached to it that's able to hydrogen bond across to the carbonyl carbon of the amide bond. So this hydrogen is able to hydrogen bond. That's also the hydrogen that's able to bond in the alpha helices. So depending on how the folding occurs, we'll have these beta sheets or alpha helices form that really help the stability of the protein. Here's just an example, more of a ball and stick form of an alpha helix, and this amino acid residues as they're turning along an alpha helix. Interestingly, you can kind of see here all of these R groups, all of these residues, they stick out the sides of the alpha helix, and they give this kind of alpha helix a flavor of how it looks. And you also look down at the beta sheets, we can see these residues stick up and down from the beta sheets, and those sheets then, these side chains are always interesting places for things to bind or interact, or other structures to interact with. So that leads us to the next structure. So this is the tertiary structure. That's when some of these residues now, right, these residues that are sticking out the side, start to interact with other residues. So tertiary protein structures, side chain interactions. So we're no longer talking about just the backbone, hydrogen bonding. Now we're talking about side chain intermolecular forces. So that's alpha helices interacting with other alpha helices, or beta sheets with other beta sheets, or alpha helices and beta sheets interacting, different loops interacting, disulfide bonds. So this is when larger groups start to interact with each other, because remember, those side chains, those residues then can stick out the side and create these more complex structures. So a lot of times, for example, channel proteins in the lipid bilayer of cell tissue is made of these alpha helix clusters. A lot of times, here's an example of mixture of alpha and beta sheets coming together to form these complex structures. A lot of times beta sheets are involved in RNA or protein or DNA, RNA or DNA binding proteins have these long groups that can interact with nucleotides as well. So these are the types of intermolecular forces that you should know. Here's, for example, ionic bonding that is shown here. This is our least favorite type of interaction, the hydrophobic. It's not really an interaction, right? It's an inclusion. Basically, these are pushed together because they don't have any of the hydrogen bonding that's going to happen with water. Here we have hydrogen bonding or dipole-dipole interactions, and this would probably be more likely or better defined as the van der Waals or London dispersion force interactions here. Disulfide bonds, we also talked about oxidation of sulfur previously, and if we have the reduced form where we have sulfur has hydrogen attached versus the oxidized form where we have disulfide bonds, we can oxidize or reduce this to break apart these or form these bonds to kind of hold these different structures together. So one of the things I want to talk about quick here is collagen. You can see these are groups of collagens, a really major important connective tissue structural protein, and a lot of these are these alpha helices that are kind of strung together of these different intertwined chains. And it also requires that we have vitamin C for the creation of this collagen residue. So these are hydroxylated, they're modified proteins. If you don't have vitamin C, this is why people get what's called scurvy. That includes joint and paint swelling, joint and pain in the joints. And this is again because we can't have that modification occurring on collagen, and it messes up with messes our collagen and the capillary walls and leads to bruising and bleeding and issues. So if you've ever heard of limey, British sailors were called this because they would add lime to their water on their ships so that they would not get scurvy because of a lack of vitamin C. Finally, we're going to talk about the quaternary structure. These are the same types of interactions, right? We still have the same types of intermolecular forces, ionic, hydrogen bonding, dipole-dipole bonds, disulfide bonds, but they're not going to be within one chain. Now we're going to be talking about combining different peptides or combining, for example, even different cofactors such as vitamin C within an enzyme. So here we have hemoglobin. This is actually multiple proteins that are bound together. So there's 4 peptides that make up hemoglobin in addition to a heme group. So these things are what's carrying the oxygen through the blood. The heme is necessary for that blood to carry oxygen because that's where the oxygen is actually binding the heme, which is also attached to the iron atom. That's important for for the function. Here you can see that this is kind of the heme and how it's kind of incorporated into this longer one of these chains of the myoglobin actually, and it's embedded in this polypeptide chain. You can kind of see this is more of a ball version of that, but this is where that heme is, and you can see the iron molecule right in the middle that's going to attach to that oxygen. So this is a quaternary protein structure, again, because we have multiple things that are attached in one. So you can see this hemoglobin. We have 1, 2, 3, 4 components. It looks just like the myoglobin, but they're all kind of attached to each other. And this is that heme group is referred to as a coenzyme. So it's a factor that's a part of an enzyme, but it's not a polypeptide. And also there's also the cofactor is like the iron ion and usually needs a coenzyme. So cofactors usually need a coenzyme to help bind. For example, like the iron needs this heme functional group. Other biochem of the cell, we focus on some of these hormones, storage proteins, transport proteins, structural proteins, right? These are more of the things that we work on in that other class. In this class, we're talking about more about enzymes and their ability to catalyze biochemical reactions. So amylase, for example, one of those digestive enzymes we recently talked about, those are the types of proteins that we're interested in for this, this class specifically. And the properties of these enzymes is that they help to help promote the breakdown or buildup of different groups of macromolecules. So hydrolysis, for example, peptide bonds, these proteins, for example, help to break down the amide bonds from long peptide chains into smaller amino acid compounds. And this is what we were talking about, where these are some of the enzymes that are released in small amounts in the stomach, also in the small intestine, that help to break down peptide chains into smaller groups that can then be absorbed by the enterocytes in the stomach. Of course, denaturation has to happen first. So this reaction, these enzymatic reactions, are breaking the primary structure. But in order, we've talked about this, most of that digestion is happening in the small intestines, mostly because that protein is all folded up and those enzymes don't have access to these folded complex forms of the peptide. So we need to heat and denature. We use acid as well to denature the protein and elongate this and break that quaternary, tertiary, and secondary structure so that the enzymes have access to these different regions. Of course, enzymes are going to only be able to recognize specific amino acid sequences in order to bind to these and break them apart. So it needs to have access to those in order for the enzyme to work to break that primary structure. So remember that most of this is happening in the stomach, and so then we're preparing that that amino acids or that protein to get digested in the stomach. Some of it happens in the stomach, but again, that's why most of it happens in the small intestines, because this has to happen before we can break it down. Other things that can influence the structure of a protein, we talked about heat. That's why when we're running gels, we heat things up, and it can weaken those side chain interactions. And mechanical agitation, also, if you kind of beat an egg white, you can get those kind of creamy. You can make different types of frostings by beating that egg, and you're actually denaturing those proteins, changing the structure. The detergents can interfere with the side chain interactions, as well as organic compounds can do this as well. pH changes in your stomach can also disrupt some of the interactions and break those different bonds apart in the different secondary, tertiary, and quaternary structures, as well as different types of high concentrations of salt can disturb those salt bridges and ionic interactions of the side chains as well. We use some of these ideas for purifying proteins out and also for running gels. That first important step, that if you're in the lab portion of the class, you notice we really need to denature. We heat those components up. We have beta-mercaptoethanol, or DTT, that helps to reduce those disulfide bonds into the cysteine residues, so that we can break apart those long folded chains into longer chains. So that's why we have that running buffer is kind of smelly, and that's why we actually heat it up at 95 degrees for 5 minutes to help break apart any of the secondary, tertiary, quaternary structures. But it should, since we don't have enzymes, we don't have peptidases in there, we should keep that primary structure intact. So we should keep that long chain. So this is essentially what we're doing. We have this folded protein with all these different side chains that have different properties. We're adding sodium dodecyl sulfate, or SDS, in there, which is kind of interacting with that backbone and then giving a general negative charge, right? This is a end of this SDS is negatively charged. It evenly coats that amino acid or that peptide, and then the electrical current helps to pull them through these gels. So these should look familiar to you, right, where we have these different markers that are going to tell us, there's standards that tell us how big in kilodaltons that protein is. Larger proteins don't run as far down this gel, so the larger proteins are going to be on top here. Smaller proteins run further down. This helps us, these ideas help us purify proteins and run them and identify them, as you've been learning in the lab. So this concludes the rest of the protein material that you're expected to know, and that is all. Thank you.